Welcome everybody. In today's video I'll be explaining mitosis. So this is a cell with a nucleus, with a nucleolus, with a centrosome, with chromatin. What is this chromatin? A chromatin is how DNA is present inside the nucleus. So this is the chromatin. Okay. After the interphase, which is like the preparation of the mitosis, instead of having 46 chromatin ropes, we will be having twice as that number. So how many? 92, because of DNA replication. I'm gonna discuss it in another video. Okay, so instead of 3, maybe I'll draw 6. Okay, and another important thing is that we will be having 2 centrosomes instead of 1 for the mitotic spindle, as we will see later on. Okay, let's begin with mitosis. So, first phase is the prophase. In this phase, what happens in this phase? This is the nucleus. Chromatin coils and condense, forming bar-like chromosomes. Look, these are the chromatin. Each one will coil like this and condense to form something like this, called chromosome. This is a chromosome that consists of 2 chromatids. Okay, so the chromosomes is like a condensed structure of chromatin. All right. And here we have something called the centromere. Centromere. Okay, so chromatin coils and condense, forming bar-like chromosomes. I will be presenting them like this, like an X. Okay, it's easier. Okay, I'll draw only 3. Each duplicated chromosome consists of 2 identical threads. As I said, these threads are the chromatids, okay? And the centromere. As the chromosomes appear, the nucleoli disappear. So let's just draw the same thing before the prophase. And what happened after or during the prophase? These chromosomes appear, and we know that the nucleoli disappear. So now I'll just erase them. Disappears. What else disappear? The nuclear envelope. So now these chromosomes will go out. But before the nuclear envelope actually breaks up, there's a really important thing that will happen. These 2 centromere will separate from each other. They will not go to the poles yet, but they will separate, and they will form the mitotic spindle. Which is microtubules. These are microtubules forming the mitotic spindle. Now, after the mitotic spindle is formed, these and the microtubules, of course, will lengthen. They will lengthen to push these centrosomes to the poles. The nuclear envelope breaks, and these chromosomes will go out to the cytoplasm. When they go out, they will allow the spindle to interact with them. So when the spindle interact with them, it will look. Some of the growing spindle microtubules attached to kinetochores, which is special protein structure at each chromosome centromere. Look, these, if I'm gonna go like this, under the microscope, same as this. But now I'm gonna highlight the centromere. This centromere that is actually connecting the two chromatids has a protein called the kinetochore. So the kinetochore. Okay, so some spindles from the mitotic spindle, okay, attach to these proteins in the centromere here, but some of them don't, like this. So the growing spindle microtubule attached to the kinetochore special protein structures, as I said, at each chromosome's centromere. These microtubules are called kinetochore microtubules. So this is kinetochore microtubules, and this is what non-kinetochore microtubules. Each will have a different function. One will lengthen and one will shorten, and we'll see now. So I'll just draw the rest. We said that we have 3 chromosomes, just in this example. Okay, so the kinetochore microtubules pull on each chromosome from both poles in a tug of war. They will pull in order that all the chromosomes will be lined on this line. Now they are not lined. Now they will be lined in what phase? In the metaphase. In the metaphase, we can see that all the chromosomes are perfectly lined at the equator of the cell, or the midline of the cell. They will cluster at the midline of the cell. And we can see also the two centrosomes are at opposite poles of the cell. And of course the spindle. Kinetochore microtubules and non-kinetochore microtubules between them, like this. Okay? So now we have enzymes that will act to separate the chromatids from each other here. So now we have enzymes that will go to the centromere and separate the chromatids. So we have, so this is a chromosome, let's say. I'm just drawing it because it's easier to understand than this maybe. So this is the centromere, right? And these are the chromatids, and this is a chromosome. These enzymes will separate the chromatid. One will go here, one will go here. So now each one of these will be separated. The two chromatids will be separated, and each one will go to one of these two poles, which later will become two separate cells after the cleavage. Okay, I will discuss it now. So, moving on to the anaphase, which is the phase of splitting. So when you hear splitting, it's the anaphase. In the anaphase begins abruptly as the centrosomes of the chromosomes split simultaneously. So they split. No connection. See, no connection. They split. But still connected to the mitotic spindle, which is connected, of course, with the centrosomes. So these are not arrows, guys. Look, this is a chromatid and this a chromatid, chromatid. Interestingly, each chromatid will become a separate chromosome. So here, after the DNA replicated in the M phase, it's just like now pinched in half. So each cell will have same amount of DNA and the same amount as the parent cell had. Okay? So there's an important thing to know, which is the different function between the kinetochore microtubules and non-kinetochore microtubules. Okay, so we said that the kinetochore microtubules are attached to the kinetochore protein, right? And the non-kinetochore microtubules are not attached to them. Okay, so these non-kinetochore ones lengthen like this, they lengthen. And the kinetochore, they will shorten. Why? Why these will shorten? Just to pull each chromatid, which is a chromosome officially after splitting, to its pole. To its pole, which will become a single cell. Okay? So look at this. We have two poles, and these chromatids will be here and here. And I will be saying now chromosomes because they are chromosomes now. So, okay, I switched cameras because there was no storage left in the first one. I was saying that moving and separating the chromosomes is helped by the fact that the chromosomes are short, compact bodies, like this, not like the chromatin, as I said. Because the fused threads of chromatin, okay, would trail, tangle, and break, so it will not work, resulting in imprecise parceling out to the daughter cells. Okay, so maybe this will have like 36 and this will get remaining chromatin. So that's why we have to condense these chromatin to form the chromosomes. Okay, without chromosomes, no mitosis. Okay, so now in the telophase, we start seeing this slight cleavage, okay? But it's not important now. In the telophase, what we have to say is that it is exactly, or somehow exactly, opposite of the prophase, okay? It begins as soon as chromosomal movement stops, because here we have in the anaphase we have chromosomal movements because of the mitotic spindle. So when this chromosomal movement stops, now telophase begins. The identical sets of chromosomes at the opposite pole of the cell begin to uncoil and resume their thread-like chromatin. Okay, so we have here coiled okay? These coiled and condensed structures will go back to its uncoiled structure. I will just erase that. Okay. Okay, so they will go back to chromatin. Okay. In the cell, where do we have these chromatin? We have them surrounded by the nuclear envelope. And what do we see next to them? We see the nucleoli and around and in the cell we can see only one centrosome. So here, each of these cells, okay, or there are not two cells now. They will be in cytokinesis, but now we say that they are one cell. So it is one binucleated cell, okay? It will form one binucleated cell after the formation of the nucleus, or the envelope of the nucleus that surrounds each group of, let's say, chromatin now, with nucleoli, and each I didn't here draw the centrosome. Each centrosome will go to a side, okay? So now you can see, if we split this cell into two cells, we will go back exactly to the first cell structure, which has 46 chromatin. And mitosis is now ended. Now cytokinesis is not part of the mitosis. The main thing that happens in cytokinesis is the cleavage furrow, which is actin filaments. It's important to know here that they are actin microfilaments, not the microtubules that were forming the mitotic spindle. Okay, so this actin microfilaments will split, and each cell will go maybe to undergo another mitosis after, of course, the interphase. It's important to know that before every mitosis we have an interphase. Okay, so just to sum up, now this is the drawing with the explanation. Now just what you need to know, the main points are this. We have prophase. What do you have to think when you hear prophase? Chromosomes formation, chromatin to chromosome, mitotic spindle between the two centrosomes, nuclear envelope, no nuclear envelope, nucleoli, no nucleoli in the prophase. Okay, they will disappear. But lately we will see that the telophase is exactly the opposite. Now, after this prophase, metaphase, chromosomes cluster at the midline, the main thing. Okay, after metaphase, anaphase, chromosomes split and begin moving. Telophase, here we have chromosomes. In the telophase, there is no chromosomes. They will go back to the DNA normal form, which is the chromatin. Mitotic spindle will disappear. Nuclear envelope will form. A nucleoli will form in the telophase, just like the opposite. And then we have the cytokinesis. Just know that there is cleavage furrow, actin filament, microfilaments, actin microfilaments, and we have two daughter cells, which are like exactly the same as the parent cell. Each one of these will undergo interphase, and then back to the prophase, metaphase, anaphase, telophase, cytokinesis. Thanks for watching. Don't forget to hit the like and subscribe for more.